Hi everyone. Can you see me, hear me? Hello, can you see me, hear me? Hi everyone. So first of all, tell me, can you see me? Can you hear me? Quickly in the chat section. Okay. So welcome to the session. Vanakkam Pasangla. Right? That's what I've learned. In Tamil, kids are Pasangla, right? So Vanakkam Pasangla, Yepidi Rukenga. I'm your biology teacher Ambika Sharma, and today we are going to continue molecular basis of inheritance. We have already discussed what is, we have already covered the structure of the DNA, right? We have already covered the structure of the DNA, right? That it is made up of nucleotides, polynucleotide chains are there. So now, today we will continue that part, okay? We will talk about the dimensions as well. Done, bache? Done? Okay. Sangeeta, creative people are usually late. है ना? So good morning, good morning, good morning. So you tell me, you revised the topic? Yes. You read it from NCERT or not? Because NCERT reading is very important, okay? So after the class, even if you are confident enough that whatever our teacher is teaching us, we are comfortable, we are understanding everything, still after the class, do read it from NCERT, okay? Because at that time, the teacher is, right? The teacher is teaching you, so teacher is explaining, so you feel like that, yes, I'm understanding everything, I know everything, but it is not always like that. After class, right? After class, do revise it from the NCERT. Fine? Ritikna, see, I think we need 3 to 4 sessions for this chapter. This is important, isn't it? Okay, so now let's continue, everyone. Let's continue. So it's molecular basis of inheritance, and you know that, right? They are Francis Watson and Crick, James Watson and Francis Crick, and here we have Rosalind Franklin, and that is this is that image of DNA, right, that she obtained after X-ray crystallography. So this can be asked as a question in your final NEET examination, right? X-ray crystallography, or it is also known as X-ray diffraction. That is the technique which is used to study the structure of the DNA. Is that clear? Okay, done, bache? So now let's continue the chapter. Okay, let's start with the bonding part first. Okay, let's start with the bonding part first. So I am going to ask few questions, so you need to answer them very quickly. Okay, so I'll ask questions, you have to answer it. So we are talking about the DNA, and we know that DNA is a polymer of nucleotide. What is it? It is the polymer of nucleotide. It's a polynucleotide chain. So here in DNA, you know that you are having purines and the pyrimidines. So quickly in the chat section, please name that purines and pyrimidines. Quickly, kindly name that purines and pyrimidines. Sangeeta, you will start soon. Yes, everyone, quickly answer it. Purines and pyrimidines, very good. So when it comes to the purines, you have adenine, you have guanine, and when it comes to the pyrimidine, you have cytosine, and then you have thymine. Okay, you have cytosine and then you have thymine. Clear, bache? Is that clear? Purines and pyrimidines are there. Now you tell me, when this purine will form bond with the sugar, then what will you call it? Yes. What will you call it? Adenine plus deoxyribose. Guys, everyone, please answer in the chat section quickly. So adenine plus deoxyribose. Quick. Adenine plus deoxyribose. It is forming? Yes, it is forming. Nandini bache, right now I am talking about the DNA. Right? I am asking the pyrimidines present in DNA. So uracil is not there because uracil is in RNA. Okay. So Vidisha, very good Vidisha, excellent. So it is going to be deoxy adenosine. Okay, it is not adenosine, it is deoxyadenosine, and it is a nucleoside. So nomenclature is important. Now, if I will add one phosphate here, yes, if I will add one phosphate here, then it will be yes, it will be nucleotide, exactly. So it is going to be deoxyadenosine monophosphate or deoxyadenylic acid, isn't it? Deoxyadenylic acid. So that's what we have covered in the DNA structure. So now you know that it is a nucleotide chain. So when you talk about that nucleotide, sugar will be there in the center. Here you have nitrogenous base and here you have the phosphate, right? Sugar, nitrogenous base, phosphate. Sugar, nitrogenous base, phosphate. Clear, bache? And you know that for making the polynucleotide chain, the sugar will join with the phosphate. Here also the sugar will join with the phosphate. Okay, so that is a polynucleotide. So here it is having phosphodiester bond, right? So this is what we have covered in the last class, that 5' end. Why are we saying it as a 5' end? See, if anyone is attending this session and if you guys have missed the last session, first of all check that session. Okay, you will understand this bonding in a better way then. Okay, so you know that sugar, phosphate, nitrogenous base is there. So when we talk about this structure, right, this sugar, its fifth carbon is having this free phosphate group. That's why it is the 5' end. Okay, bache, Vijay, that's why it is the 5' end. Vijay, you should check the last lecture before. Okay, so here it is having free phosphate group. 5' end is having free phosphate group. And when you are talking about this end, see, this is the sugar. So here 3' carbon is having free OH group. So this is the phosphate. 5' end is having free phosphate group. 3' end is having free OH group. So that is how the polynucleotide chain will form. Is that clear? That is how the polynucleotide chain will form. And I told you already that the direction will always be in 5' to 3' direction when it comes to the synthesis. Clear, bache? When it comes to the synthesis. Now, can you just tell me that length of DNA depends upon what? Yes, the length of DNA, it depends upon what? Tell me quickly. Length of DNA depends upon? Length of DNA depends upon? Yes. Number of bases or number of nucleotides? Number of nucleotides. They are going to form bases or base pairs. Number of nucleotides are going to decide the length of DNA. That's what we have studied in the last class. Yes or no? Yes or no? So that, see, it is written here now. Number of nucleotides or number of pair of nucleotide referred to as base pair, right? They are going to decide the length of the DNA. Fine. They are going to decide the length of the DNA. So once again, I'm repeating this portion, bache. This is important for you all. A bacteriophage known as this, right, known as 5 into 174 has 5386 nucleotide. Okay, bacteriophage lambda, it has 48502 base pair. E. coli has 4.6 into 10 raised to power 6 base pair, and haploid content of human DNA is 3.3 into 10 raised to power 9 base pair. This information is important. Clear, bache? This information is important, and this part we have already covered, this part as well. So it's a polynucleotide chain. 5' is having free phosphate group, 3' is having the free hydroxyl group. Now let's talk about the structure of the DNA. Okay, but when it comes to the DNA, you know that it is the nucleic acid having high molecular weight. Yes or no? What is DNA? It is a nucleic acid. So it means it is a biomacromolecule, isn't it? It's a biomacromolecule. Can I ask you one thing? Can I relate it with the biomolecules? Can you tell me one thing? Yes, can you tell me one thing? And that thing is ki, this DNA, this nucleic acid, where will you find it? In the acid soluble pool or in the acid insoluble pool? In the acid soluble pool and in the acid insoluble pool. Thank you so much, Sani. It's Salini. Thank you so much, Salini. Thank you. Tell me quickly, where will you find this nucleic acid? You are going to find it in the acid soluble pool or in the acid insoluble pool? Yes, excellent. So you are going to find it in the acid insoluble pool. If you are not understanding it, then you know what you have to do. You have to revise the biomolecule. Okay, biomolecule. So it is basically there in the retentate. Acid insoluble pool means retentate. That's what we know about the DNA, right? So now DNA is acidic, bache. DNA is negatively charged. Fine, it is negatively charged and is acidic in nature. You know that, right? It is what? It is acidic in nature, right? It is having that phosphoric acid, right? So because of that phosphate, it is negatively charged. This is important to remember. What is it? It is negatively charged because of that phosphate. Clear, bache? Clear, bache? Because of phosphate group, it is negatively charged, as well as what is happening here? It is the, yes, it is the acidic as well. What is it? It is acidic as well. Clear, bache? Clear, bache? So you know that Rosalind Franklin, that's important. Rosalind Franklin and Maurice Wilkin. Rosalind Franklin and Maurice Wilkin, they studied DNA. They studied the, or you can say that Rosalind Franklin and Maurice Wilkin, right, bache? They studied DNA with the help of X-ray crystallography, right? They studied DNA with the help of X-ray crystallography, or also known as X-ray diffraction. Again, note down. Maurice and Wilkin studied DNA with the help of, this technique is very important, X-ray crystallography, also known as X-ray diffraction. Are you getting it? Also known as X-ray diffraction. So Maurice and Wilkin, they studied DNA with the help of this. And Watson and Crick, James Watson and Francis Crick, right, in 1953, bache, in 1953, with the help of this study, they explained the structure of DNA. Okay, with the help of the study, they explained the structure of DNA. So it's not just their work, but yeah, they got the Nobel Prize. Okay, okay. You remember Chargaff rules? Do you remember Chargaff rules? Yes. Chargaff rule. Even you know that the finding of Chargaff are also used by the Watson and Crick, and with the help of that they explained this. Isn't it? With the help of that they explained it. Is that clear? Is that clear? So in 1953, Watson and Crick explained the structure of DNA, and the structure of DNA that they explained is BDNA. Okay, it is BDNA. It is right-handed DNA helix. Okay, and it's the most common type of DNA. Clear, bache? It was explained by Watson and Crick. It was right-handed. It was most common type of DNA. Clear, bache? And it was having the double strands. Okay, the DNA was helical as well as it was the double-stranded DNA. Fine, double-stranded DNA. So when we talk about this DNA, you know this thing. See. So here we know that there are two strands which are parallel. There are two strands which are parallel, but they are in opposite directions. So we use the word anti-parallel. Like this end is having, this end is 5' end. What is the meaning of being 5'? Means here that fifth carbon of the sugar is present having free phosphate group, isn't it? Fifth, 5', right? 5' carbon, 5' carbon of sugar having free phosphate group. So this side is going to be the 3' and 5' to 3' or 5' to 5' to 3'. And here it is going to be 5'. Here it is going to be 3'. Clear, bache? So both the strands, they are in opposite direction. Are you getting it? Both are in opposite direction. Let me see one strand, another strand. Okay, so if one is 5' to 3', this one will be 3' to 5'. Okay, that's how the DNA is present. Clear, bache? That's how the DNA is present. So it's double stranded. So it is made up of 2 polynucleotide chain. One is having 5' to 3' polarity. Here you will see that 5' to 3' is in the opposite direction. Is that clear? Now, bache, yesterday when we were discussing this topic, I told you already that your nitrogenous bases will be there in the center. Do you remember that? Your nitrogenous bases will be there in the center. Like this is one polynucleotide chain, this is another polynucleotide chain, and you know that it is the sugar phosphate backbone, right? Do you remember this portion? It is the sugar phosphate backbone, right? The sugar phosphate backbone is there in this DNA. It is going to support this. Remember this part? Right? Remember this part, sugar phosphate backbone is there, and these nitrogenous bases are in center. So see, these are the nitrogenous bases of one polynucleotide chain. These are the nitrogenous bases of another polynucleotide chain. So if I want to make my DNA double stranded, right? If I want to join that 2 strands, what is going to happen? Here, there will be the hydrogen bonding. What is going to happen here, bache? Hydrogen bonding will be there. This hydrogen bonding will join one strand of DNA with another. Okay, hydrogen bonding is going to join one polynucleotide chain with the another polynucleotide chain. So here, right here, Watson and Crick, they use Chargaff's rule to explain this part. Do you know about Chargaff's rule? Question can come from this part, bache. Do you know about Chargaff's rule? Tell me. Yes, I told you now that 2 polynucleotide chain, they will form bond with each other. That how will they join with each other? Their nitrogenous bases will join. They have the hydrogen bonding. Okay, so now what James Watson and Francis Crick mentioned here, that you know adenine is there. Adenine is a purine. Right, that purine will form 2 hydrogen bonds with thymine. Thymine is a pyrimidine. Okay, right? So 2 hydrogen bonds are there in adenine and thymine. It's like that. And 3 hydrogen bonds are there in guanine and cytosine. Okay, that's what we study here. Okay, that's what we study here. Adenine is forming 2 hydrogen bonds with thymine, and cytosine is forming 3 hydrogen bonds with guanine. Right? So this is what Watson and Crick explained when they were explaining the structure of the DNA. But it is all on the basis of Chargaff's rule. What this Chargaff rule is saying, as per this Chargaff rule, in the DNA, bache, amount of purines is equal to amount of pyrimidines. Right? Amount of purines, it is equal to the amount of pyrimidines. So purines means your adenine and guanine. Pyrimidines means cytosine and thymine. Right? Cytosine and thymine. So A plus G equals to C plus T. A plus G equals to C plus T. Yes or no? Yes or no? A plus G is equals to adenine plus guanine. The amount of adenine plus guanine is equals to cytosine plus thymine. Yes or no? And you know that when you talk about the concentration, quantity, this is the bracket that you use. So this is how you can write it. Fine, this is how you can write it. So can I write it in this way? Can I write it in this way? And that's equals to 1. That's equals to 1. So amount of purine is always equals to the amount of amount of pyrimidine. So in a double stranded DNA, this is going to happen. Yes, bache, this is going to happen. Now, if I talk about different species, see, base ratio can vary. If I talk about different species, see, no doubt amount of purine is equals to amount of pyrimidine, but one finding was also there that it is the adenine. Adenine is the purine which will always pair up with the thymine. It is the cytosine which will always pair up with the guanine. So obviously amount of adenine will be equals to the thymine, amount of cytosine will always be equal to the guanine. Isn't it, Tanveer bache? Isn't it, Tanveer bache? Yes, Rithigna. No doubt this is a point that purine is equals to pyrimidine, but here Watson and Crick, they also observed the complementary base pairing, right? They explained about this complementary base pairing, isn't it? Isn't it? So these were the findings of the Chargaff's rule. So adenine is pairing with the thymine, cytosine is pairing with the guanine. Okay, adenine is pairing with the thymine, cytosine is pairing with the guanine. Is that clear? Is that clear? So this is complementary base pairing observed by your Watson and Crick, and here from here, right, they also concluded from this Chargaff's rule that purine is equals to pyrimidine. So obviously it's justified, isn't it? It's justified. Okay, so now when I'm talking about the base ratio, because I know adenine will pair up with thymine, cytosine will pair up with guanine, so it is going to be, this ratio is going to be constant. Okay, this ratio is going to be constant for the species. This ratio is species specific. Okay, it is constant for a given species. It is species specific. This is what you need to remember. Okay, like we are Homo sapiens sapiens. So in our case, right, that adenine, thymine and cytosine, adenine plus thymine to the cytosine plus guanine ratio is constant for all the humans. If you talk about some other species, okay, if you talk about some other species, so in their case, this ratio is going to be constant, right? Right? They say we are humans, let's compare it, let's compare this ratio with any other animal. Okay, so in their species, this base ratio will be constant for all the members. In our species, it will be constant for all the members. Okay, so that's how it varies. This is the Chargaff's rule. Clear, bache? This is the Chargaff rule, and mainly in the eukaryotic DNA. That's what you need to remember. Mainly in the eukaryotic DNA. In eukaryotic DNA, adenine plus thymine, it is always more than guanine plus cytosine. Do you know that? Do you know that? This is the case in the eukaryotic DNA. Adenine plus thymine means the amount of adenine plus thymine is always more than the amount of the concentration of your guanine plus cytosine. Is that clear? It is always more than the guanine plus cytosine. Is that clear? Yes, bache? Quick. We have to solve some questions on the basis of that. We have to solve some questions on the basis of that. So adenine is always equals to thymine, guanine is always equals to cytosine. Okay, the base ratio, I told you that it is constant for a species. Okay, it is constant for a given species. This part. So it is constant for a given species, although it is variable in different species, but it is constant for a given species. Is that clear? These points are very important. Clear, bache? Clear, bache? Sure? Sure. So eukaryotic DNA is this: A plus T is more than G plus C. Okay, and if I talk about the melting point, you know, in our case, in our DNA, who's going to decide the melting point? Who's going to decide the melting point? G plus C. The amount of G plus C, the amount of guanine cytosine pairing, the amount of guanine and cytosine is going to decide the melting point. Is that clear? It is going to decide the melting point, or you can say that melting temperature. Fine. So melting of DNA, it depends upon its GC content. Okay, melting of DNA depends upon its GC content. Clear? Clear, bache? But overall, in our case, AT is more than GC. Fine, AT is more than GC. We'll tell you in transcription that why is it important. So this part clear, this part clear. So Watson and Crick, Because they know the Chargaff's rule, right? They observe the complementary base pairing here in the DNA, right? So adenine is forming 2 hydrogen bonds with thymine, cytosine is forming 3 hydrogen bonds with guanine. That's what they observed, right? That's what they observed. Now the next part, can you just tell me about the next part? What was next? This is what they observed now. This is what they observed now. What's next? Can you tell me about their dimensions as well? Can you just tell me about their dimensions as well? See, it's very simple. So when you talk about the DNA, okay, so when you talk about its diameter, this part, it's 2 nanometer. 2 nanometer means 20 angstroms. Okay, because 1 nanometer is equals to 10 angstroms now. So here you have 2 nanometer. So 2 into 10, 20 angstroms. So when you are talking about the diameter of DNA, it is 2 nanometer or 20 angstrom. Now, see, this is one polynucleotide chain. This is another polynucleotide chain. I told you nitrogenous bases are in center. And do you know why this pairing is like this? Why this pairing is like this? That one is adenine, another is thymine. See, it's very simple. Adenine is 9-membered ring, thymine is 6-membered ring. Now, let's say here you have cytosine. Okay, cytosine is your 6-membered ring. Guanine is your 9-membered ring. So when you talk about their structure, so somewhere, right, in the structure, like see, if purine will form bond with purine, that 9-membered ring, if it will form bond with 9-membered ring, and then 6-membered ring will form bond with 6-membered ring, the structure stability will not be there. That uniformity will not be there. So here in this case, we see adenine, purine is going to pair with the pyrimidine. Okay, like see, cytosine, 3 bonds with guanine. This part clear? This part clear? Now see, when you are talking about the single base, okay, this is the nitrogenous base, of course. This is the nitrogenous base, of course. है ना, Suhashini? It is what? It is the nitrogenous base. Yes or no? Nitrogenous base. But bache, when you are talking about this part, when adenine is paired with the thymine, then it is a base pair, right? Then you call it as base pair. If you are talking about a base, you are talking about a single base like adenine, guanine, cytosine, thymine. But if you are using the word base pair, okay, if you are using the word base pair, you are talking about a pair like adenine with thymine, cytosine with guanine. So now when you look at the structure of the DNA, see, can you see these 2 bases? One is stacking over another. So here, do you know what is the distance in between these 2 bases? Do you know? It is 0.34 nanometer, or it will be, yes, 3.4 angstrom. 3.4 angstrom. It is important. It is important, right? That's what I said. Now, DNA is a helically coiled structure. It is, Watson and Crick, they explained BDNA, right-handed DNA. It's helically coiled structure. So one base pair or one base is piling over the another one, na? Right? It is stacking over the another one. Na? So here they have this distance, 0.34 nanometer. Okay, 0.34 nanometer. So wait, I'll show you the diagram here. See this part. Look at this. It is a molecular model. Okay, so see, this is the model here. This is your DNA. So this is helically coiled. So if you will see this part, can you see this? These small spaces, this large one, this part and this part. Wait. See this. That's how the bonding is present. See, nitrogenous bases, 3 bonds. Sugar phosphate, backbone. Sugar phosphate, backbone. Is that clear? Bases are there in center. Okay, so now see this part. This is one base pair, this is another base pair. So they are having a distance of this 0.34 nanometer or 3.4 angstrom. Clear, bache? 3.4 angstrom. Is that clear? Is that clear? Yes. Is that clear? This is important. So see this part. This part here, it is the minor groove. And this part where space is more, it is the major groove. Okay, because it is helically coiled. I hope you have observed the helical staircase like this now, like this. So this part, this space is less, so it is minor groove. This space is more, it is major groove. So this complete part is forming one turn, bache. This complete part is forming one turn. Okay, so when you are talking about one turn of DNA, you will see that there are 10 base pairs. How many base pairs are there in one turn of DNA? All these are observed things. Okay, Okay, Watson and Crick, this is what they noticed in the case of BDNA. Can you see that, Nandini? So in one turn of DNA, one turn means like this, one turn, having one minor and major group. You can see this part. Okay, you can see this part. Clear? Clear, bache? You can see this part, right? So here you can see this minor group, you can see this major group. Now this is the magnified portion here. This is the magnified portion here. Major group, minor group. So this part is forming one turn. Okay, so it's understood, isn't it? It's simple mathematics that if two base pairs, they are having a distance of 0.34 nanometer, and I am saying that in one turn, if I am saying that in one turn 10 base pairs are there, right? 10 base pairs are there. So it is obvious na, it is obvious na, if two bases or two base pairs, if they are having a distance of this, so if you are going to talk about the 10 base pairs, so the distance will be 3.4 nanometer. Isn't it? The distance is going to be 3.4 nanometer, or you can say that 34 angstrom. Isn't it? 34 angstrom. Right? See, it's simple now, bache. 1 nanometer is equals to 10 angstrom. So if it is 3.4 nanometer, so 3.4 multiply by 10. So it will be 34 angstroms. Clear? Clear? So that's how you have to measure the distance. Okay, that's how you have to measure the distance as well. So same is given here. Look at this. Same is given here. So this is the one complete turn having minor groove, having major groove. Having minor groove, having major groove. So this is 3.4 nanometer. Clear, bache? This is 3.4 nanometer. Done? Done? So you should know about the units because they can ask you the question on the basis of that. Fine. On the basis of that. Done, bache? Done. So that's what Watson and Crick, they explained. Okay, so now quickly revise it from the, let's quickly revise it from the NCERT, then I will tell you about the different types of DNA as well. Okay, so see, DNA as an acidic substance, it is present in the nucleus. I told you that it was identified first in white pus cells, in the pus cells by Friedrich Mischer in 1869. Okay, so at that time he called it as nuclein, but later on Altman called it nucleic acid. Okay, Altman called it nucleic acid. So due to technical limitation in isolating such a long polymer, the elucidation of structure of DNA remained elusive for a very long period of time. At that time the techniques were not so advanced. Right? So they were not able to extract the DNA and to study that, isn't it? Right? So only in 1953, Watson and Crick, based on X-ray diffraction data produced by Wilkin and Franklin, right? They proposed a very simple but famous double helical model of structure, double helical model for the structure of DNA. So one of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chain. This was very important, the base pairing. Okay, the base pair. So this proposition was also based on, see, no doubt they explained the base pairing, but it was also based on the Irwin Chargaff, na, that double-stranded DNA will always have the ratios between adenine and thymine and cytosine and guanine are constant and equals 1. Okay, so if they explain the base pairing there, complementary base pairing there, it was also taken from Irwin Chargaff. Okay, done, bache. So base pairing confers a very unique property to the polynucleotide chain. This base pairing is providing a unique property to the polynucleotide chain. They are complementary to each other because adenine will go with the thymine only, cytosine will go with the guanine only. Clear, bache? Clear, bache? So also if each strand from a DNA act as a template, right? Like, see, this is the DNA. If one strand of DNA will act like a template, let's say it is open, so this strand will act like a template, right? It is the parental DNA. So obviously, because they explained complementary base pairing, it's understood that if we will use one strand of DNA, right, complementary to it, we can add the bases and we can form the another strands of DNA as well. Fine, Nandini, we can also form the another strand of DNA. Clear? Clear, 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 clear? Yes? So the two double-stranded DNA, okay, thus produced would be identical to the parent DNA molecule. So genetic implication. So because of this, the genetic implication of structure of DNA became very clear. So these were the points that they explained. Clear, bache? Clear, bache? right? So these are the salient features here. So you can see that it is having 2 polynucleotide chain. You know that backbone is having sugar phosphate, and the bases they project inside. So 2 chains, they have anti-parallel polarity. What is the meaning here? Anti-parallel, like see, one is like this, another is like this. Clear, bache? So bases in 2 strands are paired through hydrogen bonding. Okay, so adenine form 2 bonds with thymine. Okay, and guanine is bonded with cytosine with 3 bonds. Clear, bache? Clear, bache? So always a purine comes opposite to a pyrimidine. Always a purine comes opposite to a pyrimidine. Done, bache? So 2 chains are coiled in a right-handed fashion. So pitch of the helix is 3.4 nanometer. That pitch of the helix means actually, right, they are talking about that one complete turn. That is making the pitch of the helix. This is what you need to keep in your mind. Okay, this is what you need to keep in your mind. Clear? Pitch of the helix means that one turn. That will include that minor groove and the major groove. So the distance is 3.4 nanometer. Fine, 3.4 nanometer. And see, a nanometer is 1 billionth of a meter. That is 10 raised to power minus 9 meter. Clear, bache? Clear, bache? And there are roughly 10 base pairs. Okay, there are roughly 10 base pairs in each turn. So I just want to ask one question. If there are 10 base pairs in each turn, how many bases are there? How many bases are there? Tell me. How many bases are there? I'm saying now one turn is having 10 base pairs. So how many bases are there? 20, of course. Of course. So if one student is asking about the number of hydrogen bonds, which number of hydrogen bonds depend upon AT and CG, na? Right? If you know that how many bases are of adenine and how many bases are of cytosine, then you can answer this question. Clear? Clear? So the distance between a base pair in this helix is approximately 0.34 nanometer. So plane of one base pair stacks over the another, right? So in addition to hydrogen bond, it confers stability of the helical structure. So this is one base pair, this is another, right? And moreover, hydrogen bonding is there. That's why your DNA structure is, what? It is, yes, it is stable. Clear, bache? It is stable. So that's what you need to remember. Now see here, you have different types of DNA. It is not just the BDNA. BDNA is just the most common type, right-handed. But other than that, ADNA is also there, right? ZDNA is also there. CDNA is there. DDNA is there. Okay? DDNA is there. So here, rest all are right-handed. But this Z means zigzag. You can also see it is zigzag. So it is only the left-handed DNA. Do you know that? It is only the left-handed DNA. It is not the right-handed DNA. It is the left-handed DNA. Is that clear? What is it? It is the left-handed DNA. Okay? Okay? So that's your homework. You will write a short note on ADNA, B, C, D and Z. Okay? So Z is zigzag. It is the left-handed DNA. Fine? It is the left-handed DNA. Now, bache, see, let's say there is a DNA. DNA is having 100 nucleotides. Okay, 100 nucleotides or 100 bases are there. Okay, so out of it, your 30 are adenine. So tell me about the guanine, cytosine and thymine. Quick. Tell me about the guanine, cytosine and thymine. Quick. Just tell me quickly. Yes, बच्चे? So obviously if adenine is 30, right, thymine will also be 30. So 30 plus 30, 60. 30 plus 30, 60, right? So 40 base pairs or 40 nucleotides are left. So out of 40, 20 will be your guanine, 20 will be your cytosine. Okay, because it is a DNA, it's a double helical structure. So purine is always going to be equal to pyrimidine. Okay, purine is always equals to pyrimidine. Is that clear? Okay. So 30 plus 30, 60. So 40 are left. So divided by 2. Okay, divided by 2. So 20. So 20 guanine, 20 cytosine. 20 guanine, 20 cytosine. Okay, clear? Sure? Sure. So this is about the structure of DNA. Okay, it is about the structure of DNA and even about the configuration as well. So that's what you need to remember. Okay, done, bache. And one more thing is, yes, even if you will give your DNA a very high temperature, let's say if the temperature is above 90 degrees Celsius. Okay, if it is above 90 degrees Celsius, so in that case, what is going to happen? In that case, what is going to happen? If the temperature is 90 degrees Celsius, it is above 90 degrees Celsius. So, bache, hydrogen bonds, they break. Okay. Okay. And your two strands will get separated. So, that is known as a denaturation of DNA. What is it? It is known as denaturation of DNA. Okay, so high temperature is going to break the hydrogen bonds that will result in the denaturation of the DNA. Clear, bache? And when it comes to the renaturation of DNA, what is it? When it comes to the renaturation of DNA, obviously when the temperature is low, again hydrogen bonds will form, again the coiling of DNA will occur and you will get your DNA. Is that clear? That will be the, okay, done, bache? That will be your renaturation. Okay, Vishal, that's a good question. Guys, Vishal is asking something that where will you find zDNA? That's homework for you. Okay, tomorrow also we have a class at 10, right? So I want you people to answer this question. Clear? I want you people to answer this question. Fine? Fine? Okay, thank you, Sangeeta. Thank you so much. Fine. So the next topic that we need to start is packaging of the double helical model of DNA. Now, if I talk about the size of the nucleus, right? It is near about, your nucleus is near about this. Fine. Like the size of the nucleus is very small, but if you talk about the length of the DNA, it's too much, isn't it? The size of the nucleus is small, but when you talk about the length of the DNA, it is more. Do you know that? Approximately 10 raised to power minus 6 meter. Fine, 10 raised to power minus 6 meter. Okay, okay, so we need to understand the packaging of DNA as well. But yes, before starting this topic, let's finish this part. Fine, let's finish this part, right? Because this time we are just going with the NCERT flow. We started the DNA structure, then the salient features of the DNA, now the packaging of the DNA, then the experiments, the search for genetic material will be there, and finally we'll talk about the semi-conservative model and the DNA replication, right? So firstly, let's finish this part. So see, they have given one question here that compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? Compare the structure of purines and pyrimidines. We have done it already. Can you find out why the distance between two polynucleotide chain in DNA remains almost constant? 2 nanometer, because you know na the kind of pairing purines with pyrimidines, isn't it? Isn't it? So see, the proposition of double helical structure for DNA and its simplicity in explaining the genetic implication became revolutionary. See, what are they saying? The proposition of double helical structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology. This point is important. It is a previous year question, Pankaj. Okay, it is a previous year question. So, Francis Crick proposed the central dogma in molecular biology. Francis Crick proposed the central dogma in molecular biology. So this is important. Who proposed the central dogma? The answer is your Francis Crick. It is again, as I'm saying, it is a previous year question. What is it? It is a previous year question. Clear bache? Clear bache? So what is the central dogma of information? The central dogma of information is saying the DNA is forming the mRNA. This is how the flow of information is. The central dogma of information, it is just telling us the flow of information. So DNA will form mRNA, mRNA will form protein. This is the central dogma of information. So DNA is going to form the mRNA, that is known as transcription. mRNA is going to form the protein, that is known as translation. So it is the favorite topic of the examiner, right? They always used to ask question from this part. So this is your flow of information. Clear bache? Clear bache? Right? If you solve PYQs, you are going to find it. So DNA will replicate, right? DNA will replicate, transcription, mRNA, then translation. Very important. Fine? That's very important. Clear bache? Sure. So the next part, in some viruses, the flow of information is in reverse direction. Do you know that? In some viruses, the flow of information is in reverse direction. That is actually the reverse transcription. Do you know that? That is actually the reverse transcription. And we also call it as teminism. That is also known as teminism. What is it? Rithigna? Teminism, reverse transcription. How is it possible? It is possible with the help of the enzyme reverse transcriptase. Okay, reverse transcriptase. Clear bache? Clear bache? So here your mRNA will form the DNA, the complementary DNA. It is the mRNA that is going to form the DNA, and then the process will continue. So it is the reverse, it is the opposite, it is the reverse transcription. Okay, it is the reverse transcription. Okay, teminism it is. Okay, so in the case of retroviruses, you will see this. See, this is what they have explained, that in some viruses. Which viruses are they? Retrovirus. Why? Because these are the viruses having RNA as the genetic material. So we have the example of your HIV. Okay, coronavirus was also the RNA virus. So HIV having 2 single strands of RNA. So it is only possible because of this enzyme, reverse transcriptase. Okay, it is only possible because of the enzyme reverse transcriptase. So see, in some viruses, the flow of information is in reverse direction, that is from RNA to DNA. So can you suggest a simple name to process? Now you know that simple name as well. So see, this is the beauty of NCERT. In each and every line, there is a hidden question, and we just need to decode that. Fine. We just need to decode that. So here there are some differences in DNA and RNA, bache, right? You know about these differences. See, DNA is going to store genetic information located in the cell nucleus. It is translate genetic information into protein located outside the cell in the cytoplasm. Only present in DNA virus. DNA is only present in DNA virus. It is not present in this SARS-CoV-2. But here you know that it stores genetic information in some viruses. So I'll share the PDF, bache, right? You will get an idea of it. Now see, this is what we need to understand. Now our DNA is like this. Diameter is 2 nanometer. Okay. Now when you talk about the nucleus, if I talk about the size of this nucleus, it's 10 raised to power minus 6 meter. It's very small. If you talk about the length of the DNA, or especially if I talk about human DNA, do you know that it is 2.2 meter long? It is 2.2 meter long, and the nucleus size is this. So how are we going to fit such a long DNA molecule in this nucleus? Okay, that's a point of concern. So what can be done here? In that case, the packaging is required, and that's what we need to study. For sure, you will get one question on the basis of that. Okay, Githa, you are surely going to get one question on the basis of that. See, DNA, it is coiling. These are the nucleosomes. They are further coiling, bache. Right, they are further coiling, and this is what you are going to get, these chromatids and then the chromosome, right? So that's how it is present. So that's what we need to discuss. Okay, that's what we need to discuss. So this paragraph is very important. Okay, open your NCERT and please highlight these points. This is very important. packaging of DNA helix. What is our topic? It is packaging of DNA helix. What is it, students? Packaging of DNA helix. So you know that the distance between two base pair is 0.34 nanometer. It is 0.34 nanometer or 3.4 angstrom. So if I have to convert it into meters, it is going to be 0.34 into 10 raised to power minus 9 meter. You need to remember that. Okay. You need to remember that. 0.34 nanometer. Right? If you have to convert it into meters, that's how you're going to do. Multiply it with 10 raised to power minus 9 meter. Okay. You need to multiply it to the 10 raised to power minus 9 meter. So just look at it. If length of DNA double helix in a typical mammalian cell is calculated, if we need to calculate the length of DNA, okay, the length of DNA, which depends upon number of bases or base pairs, which depends upon number of nucleotide. So if I need to calculate that length, obviously, let's say my DNA is having 10 base pairs. Okay, 10 base pairs. So if two base pairs are having a distance of 0.34 nanometer, obviously I can check that also, na? I can check. So this is what they are explaining here. Okay, this is what they are explaining here, that if length of DNA double helix in a typical mammalian cell is calculated, right? How can you calculate it? Multiply the total number of base pair with the distance between two consecutive base pairs. Okay, okay, total number of base pairs, multiply it with the distance between two consecutive base pairs. Just in our case, in the diploid content, that's the story. In a human diploid cell, you have 6.6 into 10 raised to power 9 base pairs. You have 6.6 into 10 raised to power 9 base pairs, isn't it? So you have to multiply it with this. It is in meters. We are checking it out in meters. Okay. Okay, so 6.6 into 10 raised to power 9 base pairs, they are present in human DNA. So I need to check the length of the DNA. So I'm multiplying it with what? I'm multiplying it with 0.34 into 10 raised to power 9 meter. Okay. Clear? Clear? Yes. Right? So after calculating it, you are going to calculate it. It is 2.2 meter. Right? Human DNA is 2.2 meter. See, the information is also given for the E. coli as well. So a length that is far greater than the dimensions of a typical nucleus, which is approximately 10 raised to power minus 6 meter. How is this? How is such a long polymer packaged in a cell? So obviously here you need the coiling. So that's what you need to remember. Okay. They can right now, so you know about the human base pairs, right? That how many base pairs are there in human DNA, but in the final paper they can give you any examples. Just say, let's say if I have to change the units, I am saying that the distance between two base pairs, it is 1 meter. Imagine that. And I am saying that total number of base pairs are 100. Total number of base pairs are 100 in a DNA, and the distance between two base pair is 1. So that's how you are going to calculate it. That's how you are going to calculate it. Simple. Okay, so you need to focus on the formula. Fine, you need to focus on the formula here. Clear? So now see, this part you need to solve, that if the length of the E. coli DNA is 1.36 mm, it is mm, right? It is not meter, it is mm. 1.36 mm. Can you calculate the number of base pairs in E. coli? Yes, can you do that? You need to use this formula, of course. Right? You need to use this formula. Can you explain that? Can you tell me that, bache? And I'll ask one more question from you. I have one question. I will ask it. So tell me. Tell me about it. Give it a try. Give it a try. Anyone in the class? Anyone in the class? 1.36 mm is the length. Actually, the bases are 4.6 into 10 raised to power 6 base pairs. So now you have to check how is it possible. Okay, this is what you need to check. Fine, this is what you need to check. So it is 4.6 into 10 raised to power 6 base pairs. Okay, it is 4.6 into 10 raised to power 6 base pairs. Clear, bache? Fine. Okay, so before going forward, I will ask you one question. Okay, can I ask you one question? Will you give it a try? The question is, you have to calculate the number of nucleotides. What you need to do? You need to calculate the number of nucleotides in a DNA. That's a very small fragment of DNA. You have to calculate the number of nucleotides. Okay, okay. So this DNA is having 80 thymine. This DNA is having 80 thymine, and it is having, see, 80 thymine and 80 guanine bases are there. So you need to calculate the number of nucleotide. Can you do it? Yes, give it a try. Give it a try. Quick. Give it a try. Bache, it's 6.6 into 10 raised to power 9 base pair, na? But when you are converting 0.34 nanometer into meters, then you are taking 0.34 into 10 raised to power minus 9 meter. Bache, Vela, I think there is some confusion. So, bache, can you just calculate it? You have to calculate the number of nucleotides, right? You know about the thymine, you know about the guanine. How will you do it? Give it a try. Give it a try, quick. It's very simple. It's actually very simple. Simple mathematics. It's simple mathematics. Very good, Nandini. Very good. See, it's very simple. Number of nucleotides you have to check now. So if you have 80 thymine, that means you have 80 adenine as well. 80 plus 80 is equals to 160. Same way, 80 guanine means 80 cytosine, again 160. Now you have to check the number of nucleotides here. Total number of nucleotide means total number of bases. These are the base pairs. No. So 160 plus 160. 320 it will be. It's very easy. है ना? It's very easy. That's how you have to calculate. Fine. Okay. So now let's talk about the packaging of the DNA. So first of all, let's talk about the prokaryotes. Now when we talk about the prokaryotic cell, okay, so in their case, we know that the genetic material is DNA is circular. It is there in the cytoplasm. DNA is circular. It is there in the cytoplasm. Yes or no? Your DNA, it is circular, it is there in the cytoplasm. We know it very well. And this DNA, we use the word nucleoid for it or the genophore for it. Okay, genophore for it. We used to call it nucleoid or we used to call it genophore. What we used to call it? We used to call it nucleoid, we used to call it genophore. Is that clear? Is that clear? Here you have the cytoplasm. Here this is what your genetic material is. You are calling it as nucleoid or genophore. Actually, what is happening when you are talking about a prokaryotic cell, we know that there is no nucleus there in the prokaryotic cell. Genetic material will directly be there in the cytoplasm. Moreover, here, no doubt, even the prokaryotic DNA is highly coiled, but histones are absent in prokaryotes. Okay, but histones are absent in prokaryotes. Clear? Here you will see some other proteins, some other basic proteins. They are polyamines. What are they? In the prokaryotes, polyamines are present. These are some basic proteins, positively charged proteins. So your DNA will coil over that polyamines, right? And you will see the coiled genophore or the nucleoid here. So what is the meaning of nucleoid? devoid. Devoid of what? Devoid of histone proteins. It is actually the naked DNA, not present in the nucleus and not covered with the histone proteins. Is that clear? Right? So here for the coiling, positively charged polyamines are present. Fine. Positively charged polyamines are present. Clear, bache? Next is your eukaryotic DNA. Next is your eukaryotic DNA. Here you have the nucleus, right, which is 10 raised to power minus 6 meter. Okay, so here you have to pack the DNA which is 2.2 meter long. So we will go for the packaging. Okay, and the very important thing here is the histone proteins, right? Histone proteins. See, bache, your DNA is negatively charged, isn't it? DNA is acidic and DNA is negatively charged. DNA is acidic, DNA is negatively charged. Now what is happening here? What exactly is happening here? We have some proteins, histone proteins. Which proteins are they? They are your histone proteins. But these are basic proteins. Basic proteins means they are positively charged proteins. This is the first level of coiling. Okay, they are positively charged proteins. They are basic proteins actually. Okay, they are basic proteins actually. They are the positively charged protein. So what is the point here? Okay, what is the point here? As I said, histone, basic, they are what? They are the positively charged protein. Okay, they are the positively charged protein. Fine. Now what's the point here? What is the point here? Why these proteins are positively charged? Because they are made up of the basic amino acid. So it is a previous year question, bache. Just a minute. It is a previous year question as well. Ishwat, it is a previous year question as well. Histone is made up of which amino acid? In the histone, your lysine and arginine, they are present. This is an important question. Which amino acids are making your histone proteins? Lysine and the arginine. You know that basic protein means having basic amino acid, and proteins are the polymers of amino acid. Many amino acids, they will join together and they are going to form that protein. So lysine, arginine, so many lysine and arginine, they are forming these histone proteins. Okay, and yes, just for your information, this is the trick for the basic amino acid. It's whole histidine, arginine, lysine. Okay, these are your basic amino acids. But in the case of histone proteins, it is lysine and arginine which is present. Fine, Ishwat, it is lysine and arginine which is present. So we have what? Histone proteins are there. So it is the first level of coiling. What is it? It is the first level of, yes, it is the first level of coiling here, right? So it is known as nucleosome model. It is the nucleosome model. What's that? It is the nucleosome model, isn't it? Isn't it? So we are going to talk about the histone proteins here. So which histone proteins are there, bache? It is H1, H2A, H2B, H3, H4. Right? That's what you need to remember. What do you have here, Soumyadeep? H1, H2A, H2B, H3, H4. These five, okay, these five histone proteins are present. These five histone proteins, right? These five histone proteins are present. They are involved in this nucleosome model. So what is happening actually? Bache, see, your H2A, H2B, H3, H4, they are forming an octamer. This is important. This is important. Okay, see, it is a nucleosome model. Actually, what is a nucleosome? When the DNA is coiled over these histone protein, when DNA is coiled over the octamer, this octamer of histone proteins, right, then you call it as nucleosome. But before that, this is what you need to understand. This octamer is made up of these four proteins, two copies of H2A, 2 copies of H2B, 2 copies of H3, 2 copies of H4, right? So we have four proteins. Their two copies means total 8 proteins will be there, right? It will be total 8 in number. So that is why you are saying it as octamer. That's why you are saying it is an octamer. Is that clear? Is that clear? Yes. Is that clear? So histones are organized. They will form a unit of 8 molecules which is known as histone octamer. Clear bache? Histone octamer. Done bache? So it is positively charged. So now the DNA will coil over it. DNA which is negatively charged, it will coil over it. Now you are calling it as nucleosome. So this octamer here, right? This octamer here is what? It is also known as new body, right? It is also known as new body. This is the octamer of the histone proteins, new body. And when this nucleosome, when this DNA coils over this core of the nucleosome, or when this DNA coil over this octamer, now you are calling it as nucleosome. And this nucleosome contains near about 200 base pairs of DNA, imagine. Right? It contains near about 200 base pairs of DNA. So that's how the coiling will occur. This is the first stage, the nucleosome model. Okay. And yes, bache, you have, see, H2A is involved, H2B, H3, 4. They are involved in the octamer formation. They will form the octamer. Octa means 8. So these 4 proteins, they will form, right? They are forming this octamer. Now, what is the role of this H1? H1 is basically the linker histone proteins. Just see, right? It is not only one. It is not like that, bache, that only one nucleosome will form. No, that's how the coiling will occur. That's how the coiling will occur. And see, this part here, this DNA here is the linker DNA. What type of DNA it is? It is the linker DNA. What type of DNA it is? It is the linker DNA, isn't it? Isn't it? So to this nucleosome, here you have your H1, the linker histone protein. So this is the linker histone protein associated with your linker DNA. This is making the complete nucleosome. Okay, it is making the complete nucleosome. So that's what you need to remember. So they can ask you the question, which histone proteins are playing role in the packaging of the DNA molecule? Which one is acting as the linker? Which one is making the octamer? So such questions can come in your final exam. Okay. So this is one nucleosome, this is another. Roughly they are acquiring 200 base pairs, and in between this DNA is your linker DNA. Right? This DNA is your linker DNA. Is that clear? Sure? Are you sure? So just see this diagram. Important it is. So this is the core of histone molecules. Here you can see the DNA, and here you can see H1 histone. Here you can see H1 histone. So bache, when you see it under electron, EM picture. EM picture means which microscope it is? Do you know about this? It is written as the EM picture. Electron microscope it is. Okay. So under the electron microscope, it appears like this. Beads on a string. Beads on a string. So that string part in between beads is actually that linker DNA. So these are beads on a string. That's how it looks under the microscope. Okay, under the microscope. Is that clear? So a typical nucleosome contains 200 base pairs of DNA helix. A typical nucleosome contains 200 base pairs of double helix. A typical nucleosome contains 200 base pairs of DNA helix. Clear, bache? Okay, that's important. Fine, that's important. Okay, so nucleosome constitute the repeating unit of structure in nucleus, right? These nucleosomes, now they constitutes the repeated structures of this DNA in that nucleus. Now they will form chromatin. Okay, now see, this is how the coiling will occur. Okay, so now what will they form? When your DNA is coiled over the histone proteins, you use the word chromatin. You know this word, na? Chromatin. Chroma means color. It can be stained with the basic dye. So it is the chromatin now. What is it, bache? It is the chromatid. So now it is thread-like. Okay. Okay. So nucleosome and chromatin, they are basically seen as bead on string. Fine. They are basically seen as beads on string. Done, bache? Okay, sure. Sure. So, bache, do you know the diameter of your nucleosome? Yes. Do you know the diameter of your nucleosome? It is near about 100 angstrom. See, we started the story from 20 angstroms. Isn't it? We started the story from 20 angstroms. Now here, the diameter of nucleosome is near about 100 angstroms. And you know, in the human DNA, right? Human DNA is 2.2 meter long, right? So in human DNA, near about 33 million nucleosomes are present. Okay, 33 million nucleosomes are present when it comes to the human DNA. So imagine the level of packaging. Clear bache? Clear bache? Imagine the level of packaging. Fine. So first is the nucleosome model. First is the nucleosome model. So next is your solenoid model. These are just the level of packaging. Okay. That first of all, nucleosome they need to form, then that repeated units, that nucleosome consists of that chromatin. Now the solenoid model, like this, that nucleosome, let's say if it was like this, so it will further coil. It will further coil like a solenoid wire. And now you know, when it comes to that solenoid, what is the diameter there? It will be near about 300 angstroms. Imagine. It will be near about 300 angstroms. So these are just the level of packagings. Clear, bache? These are just the level of packaging. So obviously you need the proteins there, isn't it? You need the proteins there. Clear, bache? Clear, bache? Okay. So next is solenoid model, and then comes the radial loop model, right? These are the levels of packaging. Like this solenoid will further coil. Wait. See this part. See this diagram. 2 nanometer. It is further coiling with histone proteins. Okay. So if I see in the case of nanometer, it is 11 nanometer. If I talk about the in angstrom 20, here it is near about 100. So here they have given it in the form of nanometers. 11 nanometer. Then it is further coiling. So you can see this, right? They are forming the solenoid, right? So here the diameter is 30 nanometer. The solenoid is having a diameter of 30 nanometer. It is further coil, right? So this is next level of packaging, that is radial loop model. This solenoid will further pack. Okay, this is, suppose this is your DNA, right? You are coiling it, right? You are forming the nucleosome chromatin. You are further coiling it. You are forming solenoid. You are further coiling it. You are forming what? You are forming that radial loops. Further, after coiling, you will form the chromatid. The chromosome. Clear, bache? Clear, bache? So see, 30 nanometer, then 300 nanometer, right? These are the radial loops, na? Their diameter is 300 nanometer. So further it will coil, and you will get your chromosome 700 nanometer. So it is the chromosome before your S phase. So after S phase, this chromosome is total 1400 nanometer. So imagine we started the story from 2 nanometer. Look at the level of packaging here. Okay, look at the level of packaging here. Is that clear? Yes, bache, that's important as well. Right, 2, then 11, then 30, then 300, then 700. Clear? And finally this chromosome having this 1400 nanometer. Is that clear? So initially initially histones are required, then bache some NHC are required, right? Some NHC proteins are required. These are another proteins. Wait, see this. Come to this part. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered. You know that, right? It is forming the nucleoid. It is held with some proteins, and I told you that proteins are your polyamines. They are forming the nucleoids. So in the case of prokaryotes, DNA in nucleoid is organized in large loops held by proteins. Okay, held by proteins. Now next part. In eukaryotes, this organization is much more complex. So there is a set of positively charged basic proteins called histones. There is a set of positively charged basic proteins called histone. So protein, they acquire charge because they have a basic amino acid. You know that lysine and arginine it is. Okay. Okay, bache. So both the amino acid residue carry positive charge in their side chain. Histones are organized to form histone octamer. The DNA will further coil it over. It will form nucleosome. So typical nucleosome contain 200 base pair of your DNA helix. Important information, right? 200 base pairs of DNA helix. And then nucleosomes constitute the repeating units of structure in nucleus called chromatin. Clear? So nucleosome, they constitute the repeating unit of structure in nucleus. This is the chromatin, which is thread-like, bache. It looks like beads on string. Fine? Beads on string. So beads on string structure in chromatin is packed to form further chromatin fibers that will further coil and condense at metaphase stage of cell division. So you know this portion, right? Right? So you can use that image. You can just note down the dimensions as well. Okay, so this image is important. So as I said, for the next level of packaging, you need non-histone chromosomal proteins, NHCs, non-histone chromosomal proteins. So some region of chromatin, they are loosely packed, bache. For next level of packaging, like I told you, histones are there. Further, you need to coil it. So non-histone chromosomal proteins will be used there. Fine. So in a typical nucleus, some region of chromatin are loosely packed. It stains light. Some regions are loosely packed. Just say, for an example, when I was showing you the linker DNA, so that DNA is free. That is loosely packed. So it is available for transcription, right? We can use these genes. We can use these genes to make the proteins. It is available for transcription. We can use such genes for making proteins, so it is available for transcription. So such type of DNA is your euchromatin, true chromatin. Okay, it's loosely packed, it's lightly stained. Why we are saying that it is a euchromatin? Because this DNA is available for transcription. We can use the information of this DNA. We can decode this DNA. Okay. So when it comes to the heterochromatin, so when DNA is very, it's compactly packed, or nucleosome where DNA is compactly packed, that DNA is not available for transcription. It is transcriptionally inactive. So now we have just studied the central dogma of information, isn't it? So we know that DNA will form mRNA, mRNA will form protein. These are the enzymes important for metabolism. Clear? So no such thing will happen here now, isn't it? So DNA will form mRNA, mRNA will form protein. Are you getting it? But here this DNA is not available for the transcription. It is not available for the transcription. It is not available for the formation of the mRNA and the protein. So it is heterochromatin, darkly stained, transcriptionally inactive it is. Clear, bache? So bache, earlier I was thinking that I will even teach you the experiments today, but that's not possible. So that we will discuss tomorrow, right? Tomorrow again at same time we have a class. So we will discuss it then. And then after experiments, you know that each class is required, because see this, like I will try to stretch your classes for the molecular basis of inheritance. Okay, I will try to keep them for 3 to 4 hours. See, we are doing this chapter for the first time on this channel, Unacademy NEET English. Okay, so I want to deliver you that quality content. Later on we will start one shots where it will be a quick revision. Okay, but as of now, that's important for you. You should understand each and every line of NCERT. So proceed accordingly. Clear? So I'll share the PDF in the Telegram group. Don't worry. So start revising it. And if you are new to our channel, what you have to do? You have to subscribe to it. Okay? And join the Avengers batch. Soon we are going to start neural control and coordination in human physiology. And you will get sufficient time for the quick revision as well. Right? Don't worry about it. You will get the time for the revision as well, for completing your backlogs. You will get the revision classes also. Okay? So take care everyone. Bye-bye.